Hi dear students, a warm good morning to all of you. Let's continue with infections. And as we all know, infection, a chain of infection mainly involves different types of factors, and the success of infection or the progression of an infection mainly involves the combinatorial or the sequential activity of all these factors. Let it be a pathogen which causes an infection, the virulence factors of the pathogen, the exposure time, the dosage of the infectious agent, as well as the susceptibility of the host to a infectious agents. These are all things which contribute to the chain of infection. And if any one of this chain is being interrupted, the progression of the infection is being disrupted. So today we are going to talk about the virulence factors which mainly contribute towards the chain of infections. When we talk about the virulence, we also need to talk about the term pathogenesis. What is pathogenesis and what is virulence? Both of them seem to be related to each other, but there do exist some differences. If you take an infectious agent, the ability of the infectious agent to cause disease is referred to as pathogenicity. And usually the pathogenicity is considered to be as a property of a particular bacterial species or its ability to cause a particular disease. But sometimes you can see that not all species or not all bacterial species will be found to be pathogenic. And also, there also do exist that among the species of a bacteria, different strains of bacteria do have ability to cause disease at a different extent. That is, depending upon the strain, the ability to cause the severity of a disease is being found to be different in different strains of a particular species. And that is mainly attributed to the term of virulence. So virulence, it specifically refers to the magnitude of pathogenicity caused by a particular microorganism. And it is also determined by the degree to which the pathogen causes damage, including invasiveness as well as infectivity. So if you go to consider pathogenesis as a property of a microbial species to cause disease, the virulence is attributed to the strain of a particular species. More specifically, let's observe. If you go to take virulence, virulence is a measurement of the ability of a disease, of a strain to cause disease in an organism, whereas pathogenicity refers to as the ability to cause a disease in the host organism by a particular species of a microbe. And virulence is a quantitative measure, whereas pathogenicity is considered to be as a qualitative measure. Pathogenicity just implies whether the organism can cause disease or not. But how extent or to what extent different the pathogenicity or the harm or the infection can be caused, that is mainly related to the virulence. So virulence is mainly used to express the degree of harmfulness of a particular pathogen, and but pathogenicity is not suitable to explain the degree of harmfulness of a particular pathogen. In spite of all these differences which we mark, we also note that there do exist some similarities between pathogenesis and virulence. They are sometimes considered to be as interchangeable terms in some extent, and both of them, it mainly they refer to the ability of the pathogen to cause a particular disease. But we can see that pathogenicity depends upon virulence. And virulence and pathogenicity have a different genetic control when we go into the depth of it. Now, in this section, we are starting as what are the factors which mainly contribute towards the virulence of a particular organism. Of course, the virulence means the ability of a particular strain to cause pathogenesis or to increase or the severity of the disease which is being caused by a strain is referred to as the virulence. Now, this depends upon various factors. It could be the factors which are attributed by the organism, or it could be by the factors which have been attributed by the host also. Often you can see that there occurs a chain of interactions which would happen between the host and the pathogen. The host susceptibility is something which will influence the virulence, and the host pathogen interactions will further control it. So there are some host susceptibility factors and host pathogen interaction factors which will contribute to the what virulence of a particular pathogen. But in this session right now, I am just concentrating on the factors which are inbuilt within the pathogen and which enable it to cause pathogenicity or virulence. So among these factors, on the surface, I'm not going to talk about the host factors right now. We are concentrating on the pathogen-based factors. If you go to consider the pathogen, there are various components on its surface as well as within it, which will enable it for its virulence. Now, sometimes it could be surface molecules which have been attached to it, like we have fimbriae, then we have siderophores. Sometimes siderophores are ion chelating substances. Sometimes you might have toxins which have been produced by the bacteria, like enterotoxins, or which are involved in diarrhea, or it could be endotoxins, okay, which has been found to be intracellular, or sometimes there might be LPS or lipopolysaccharides involved, or antiphagocytic proteins which have been produced by the bacteria that would also be substances which are responsible for its virulence, or on the surface of it, on the surface of the particle, there would be certain antigens which will inhibit phagocytosis. So that could contribute to its virulence, or it could be some flagella like substances which will enable it for adherence or stick onto the particular cell. Then we have capsules which will inhibit the complement binding mediated emission of the bacteria. Then there could be various other cytotoxins. So there are a wide range of factors produced by the bacteria and which is responsible for its virulence also. So we are not going into the full depth of what are the factors which are responsible, and but a overall view is being done on the various factors which are responsible for the virulence. And in this figure also you can see that there are certain plasmids which have been found in certain bacteria which is responsible for virulence also. So let's move on. Firstly, let's talk onto the pathogenic islands. Now, what are pathogenic islands? If you go to look onto certain pathogens, you can see that they have large segments of bacterial chromosome as well as plasmid DNA, which have been found to encode a virulence factors. And these factors, these segments are referred to as the pathogenic islands. And they appear to have inserted into the existing DNA. Now, if you go to consider Yersinia species, Pseudomonas aerogenes, Ashigella, Flexneri, Salmonella species, etc., they will carry at least one pathogenic island. And the pathogenicity islands generally increase the bacterial virulence and are absent in the non-pathogenic members of some genus as well as species. So if you go to look onto different bacteria, you can see that they have different pathogenic elements, pathogenicity islands. What are pathogenicity islands? These are just genetic elements which are being present either in bacterial form, bacterial chromosomal form, or in bacterial plasmid form. And these are these are in fact bases, nucleotide bases, which will code for certain substances, which will increase the virulence of the particular substance. Now, what does they encode? It depends upon different organism. Sometimes they might produce certain secretion proteins which would serve as cytotoxins. Sometimes they would serve as pile-secreted protein which will help in attachment, or cytotoxic functioning proteins would be there, or sometimes there would be substances which will enable them for the intracellular survival, or some would be toxins, or some would be enabling them to take iron, or sometimes some components which, as we have you can see in Staphylococcus aureus, the pathogenicity island will code for a protein which will serve as a super antigen. So the presence of certain genetic elements, like of about 100 to 200 base per kilo basis of DNA, sometimes would code for the virulence factors in different organisms, and they are referred to as the pathogenicity islands. Now, other than the genetic elements, if you go to look into the bacteria, you can see that the property or the events of addition, colonization, as well as invasion plays an important role in its virulence. Now, what do you mean by addition? Addition mainly refers towards the attachment of a bacteria onto a particular host cell, and all those substances which will enable it for addition is referred to as adhesins. Now, adhesins could be of different types. They could be either be well organized structures such as pili or fimbriae, or they could also serve as colonization factors. Now, in the absence of adhesins, the strains become avirulent, or they lose their virulence. And you can see that certain specific adhesion molecules might be involved when the bacteria have to show a particular tropism to a tissue, that is, or a specific attachment of a bacteria to a tissue might sometimes involve specific adhesins. And the host specificity is also being exhibited by certain specific adhesins. Now, what are these adhesion molecules? As I told you, they could be either be well organized structures like pili or fimbriae, or sometimes there would be other certain specialized proteins also, depending upon the what the main purpose of an adhesion is, just to attach onto the particular host cell. So different bacteria have different adhesion molecules. Let's just have a look. Now, there are different mechanisms, as I told you. Sometimes fimbriae, which have been present on the particular bacteria, will enable the bacteria to attach, or sometimes even certain particles which have been present on the capsules, okay, that will enable. And now in the case of viruses, certain proteins or certain spike proteins which have been found onto them, onto the surface of the virus, would also help in the addition or the adherence. So these are the different mechanisms, and these factors, these adhesins, which will help them to attach onto the particular host cell, would also serve as virulence factors in them. After the process of addition, of course, the host onto the host cell, they also need to colonize. Now, what do you mean by colonization? Colonization mainly means the establishment of a site of microbial replication within on or within the particular host. And it might not does not need to lead on to tissue invasion or damage, but it involves that after attachment, the microbe should be able to what develop a site of microbial replication over there. And it depends upon the ability of the pathogen to survive in the new host environment and to compete successfully with the host cells and the host normal microbiota for the essential nutrients. And you can see that specialized structures would also, which enable for the host attachment, is also needed for colonization. So the factors which have been responsible for what do you say for adherence could also contribute to colonization. After colonizing, the bacteria has to invade the tissues. Now, what do you mean by invasion? The invasion means the ability of a pathogen to spread into the host tissues after establishing the infection. And there could be different types of organisms, like there would be highly invasive strains like Streptococcus septicemia, which is being found in wound infection. That is a highly invasive pathogen involved. Or there could be less invasive pathogens which causes localized lesions. Now, for example, Staphylococcus abscess. And some pathogens can cause serious diseases, might in fact lack invasiveness altogether. Now, for example, tetanus bacilli that is confined to the point of entry. So now you might think, what is the difference between all these? Now, whatever be the mechanism by which the organism enters into a host cell, it it might either involve to invade a particular host. It will have to break in and go to the surrounding tissues. When an organism just enters into it, that is what it follows a process of addition as well as colonization. And when it tries to expand towards the surrounding cells, that refers towards invasion. And the process of invasion mainly involves the movement of the microbe from one cell to different cell or to different tissues. Now, if it's penetrating through the host membrane or epithelium, it can do it through active mechanisms or passive mechanisms, and then it will move towards throughout the particular host. And sometimes you can see that they might enter through the lymphatic system, and from the lymphatic vessels they might enter into the circulatory system, and from the circulatory system you can see that the bacteria might be in the circulatory system itself. It might be spreading throughout the bloodstream, and that is referred to as bacteremia. And sometimes even the products or the toxins produced by this bacteria as well as by the fungi might be found to be present in the blood, and that is what we refer to as septicemia. So whatever be the mode of invasiveness, you can see that uh, various factors are responsible for it to penetrate into the different membranes of a particular cell and spread from one host tissue to the one tissue to the next tissue. And these factors, which will help to increase the invasiveness, are also virulence factors. Now, you can see that different organisms have different factors, virulence factors, which enable them for invasion as well as dissemination. Now, in coagulase, that is a particular protein produced by Staphylococcus aureus, it will what? Protect the pathogen from phagocytosis. It will coagulate the clots of the fibrinogen. These are all enabling it for invasion. Clostridium species has collagenase enzymes which will break down the collagen. Then some bacteria have enzymes such as elastases and alkaline proteases which have been found in Pseudomonas aeruginosa. Sometimes there would be hemolysins. Hemolysins are which will help to lyse the erythrocyte. That is, if it has to enter into the RBC, it will penetrate the hemolysin. Okay. Sometimes there would be high hyaluronidases, which will help to break down high hyaluronic acid, which is mainly being found in the extracellular matrix. So depending upon where the organism has to invade, it will produce different types of virulence factors, and they are inbuilt within the organism, and they promote the what these are virulence factors, and these virulence factors contribute towards its invasion ability as well as its virulence ability. So let me just try to help you to distinguish between what is adhesion, colonization, and invasion. If you go to look into this cell, the prokaryotic cell over here mainly refers towards a bacteria by various factors like addition factors like pili, capsule, invasions, toxins, etc. There are different things over here. Using different adhesive factors, the bacteria can attach onto the particular cell. Okay, and the colonization mainly involves, instead of one bacteria attaching, many bacteria together attach onto the particular cell and form a colony over here, or they establish a local environment for their growth. That is referred to as colonization. After that, if the bacteria enters into the cell or progresses from one eukaryotic cell into the next cell, that refers to the process of invasion. And all these factors which are responsible for either addition, colonization as well as invasion are all examples of virulence factors which contribute to the virulence of a particular pathogen. And if you use any mechanisms to block either of these, the infection can be controlled. So I guess it is clear to you. We will be continuing with the remaining virulence factors in the next session. Thank you for now.